Yeah, structural variants are uh, genomic alterations of like roughly 50 base pairs or longer, and they fall into like 5 basic categories: insertions, deletions, inversions, duplications, and translocations. And in many cases, there are like complex rearrangements that are a combination of 2 or more of these. In this case, or for my talk, duplications and deletions will also be counted as copy number variations, that they will affect the copy number status of like a gene locus or like a genomic loci. And finally, this structural variance causes a higher diversity in human genomes and genomes in general if we compare the number of bases that are covered. So here are some illustrations of all the structural variance that exists. And when we use long-read technology, we have like a more kind of comprehensive way to study structural variance. First, we have not only a higher number of them. So talking about human genomes, if we are using long-read technology, we have almost like double the number of structural variants that we detect. But not only that, we also can access some regions that are difficult for short-read technologies. One of such cases is this 193 medical relevant challenging genes that like contain like highly repetitive kind of sequences that are hard to assess with short-read technology. And also with long reads, we can assess centromeric and telomeric regions. So kind of this is a roadmap of like all variations in human genome, how to detect them. So for short, like small deletion variants, we have SNVs, so we'll not talk about that. And for structural variation, we have Sniffles. So we are kind of aiming on the range of like 50 to like MB sizes. And with Sniffles 2, our current version that we're going to release, 2.2, we can detect germline structural variation, fully genotyped population datasets in a matter of seconds. Also detection of low frequency, somatic or mosaic structural variants. And we have included now what we call a cancer pipeline to be used like either combining the mosaic and the germline calling with the population merge in order to have a more comprehensive view of a tumor-normal comparison. But that's not the only thing. We also have a new tool for CMV calling for long reads called Spectra. And we can use this to assess like megabase and larger like copy number variations. We can, basically it's based on coverage, but you can use SNVs to improve the calling, and also we are aiming also to do LOH. So the copy number status can be like neutral, but you can have loss of heterozygosity. Also, we're working similar steps, Sniffles, to have the fully genotyped family and population calls, and we have an experimental approach for still like work in progress for cancer, as these genomes tend to be really, really like messed up, a lot of aberrations. So estimating the correct like copy number status as neutral it's been challenging for us. So first, I'm going to talk about Sniffles. First, we benchmark it with currently available tools at the moment, QSV, PBSV, Sniffles 1, and SVIM. And we use for that the HG002 gene amenable benchmark, both the tier 1 and the challenging medical relevant genes. And as I can show you here, Sniffles 2 is the kind of the best performing. Like in this case, like all the blue figures are in default mode for all the tools. All the red ones are when we tune the parameters for all the competitive tools on a wide range of coverages from 5 to 50x. And as you can see by the genotype F1 score, Sniffles 2, when compared to other tools, has the best performance. As you can see here, the stars are not even here because the genotype F1 is below 0.5. And time-wise, Sniffles 2 is quite fast. So this is the time in CPU minutes. So you can see that for like here, some case, we have here for 10x for QSV, and that's the same time that it takes like 50x sample for Sniffles 2. So yeah, very impressive. We can do germline calling, but we're interested in like using our tool for like a new kind of discovery of structural variant. And one of the things that we develop is the population merge. So basically, you touch your BAM file once, and then you forget about it. So for each sample, we not only produce a BCF file, but also a binary file. It's roughly like 10 megabytes. And it contains all the candidate structural variants. So when you need to merge files, you can use only this binary file in order to produce a fully genotyped population BCF in a matter of seconds for a family trio. And when you add samples, it almost linearly scales. We have seen this shown at the G42 talk. Yesterday, like if you ramp it up like the thousands, it would just take more time. But we're working on improving this time. And finally, when you compare or merge against like a naive approach, like calling structure variants for each of the files, have all the BCF using Survivor, you will have a lot of genotypes that will be missing. And then in order to remove this missingness or to improve, you need to regenotype all the kind of samples by like using again the BAM file. So that takes a lot of time. And still, you can see that for Sniffles 2, the number of like NAs that we have is way smaller than for the force calling using QSV. So we applied this population merge to detect structural variants that were disease relevant in the MECP2 locus. We had a population of 31 individuals, some of which were affected, and we were able to detect structural variants that were unique for each of these individuals. And these structural variants mostly affected the gene dosage, which is important for this gene. This is one of such examples. So we didn't do all the job with Sniffles 2, but we have like array data from our collaborators in order to have the complete kind of order of like how this big arrangement is. So we have here like the duplication, then segmental from the gene, the locus, and we have segmental duplications here, which affected the array. Then have a triplication and duplication again. And the Sniffles 2 was able to detect an inversion, and with all this information together, we managed to get like the whole resolution of the event. We're applying this population merge to large cohorts, for example, in Gregor, but it can also be applied like for like larger, as the G42 people like mentioned it yesterday, and also to compare within like samples, for example, in the brain that Kim just mentioned before. So the next part is the Mosaic structure variant calling. So in this case, I'm showing you an example of a low frequency HG002 that was mixed on a higher frequency HG073. This is in order to simulate what like mosaicism would be. And what you can see, we can detect like a couple of these like insertions. So how do we perform on that regard? So we tested QSV and Sniffles 2 in default mode, and you can see the precision recall are pretty bad. And when we use our Mosaic mode identify structure variants up to like 2 or 3 reads, you can see that the precision went up when the proportion of structure like the HG002 was up. But weirdly enough, we see like this trend in which at like 7% proportion of HG002, the recall was really bad, and we saw like why is it happening? And the thing is, Sniffles 2 only detects structural variants within the 5 and 20% allele frequency, so everything else is just not taken into account. When we just focus on that part, we can see that the recall on that proportion is quite high. And we applied this to an MSA sample that's a rare neurodegenerative disease with Parkinson-like symptoms. And we can show you here, we have here ONT bulk, Illumina bulk, and Illumina single-cell sequencing. And we were able to detect this deletion in an important gene. And there's some of the single-cell sequencing from Illumina also kind of show this. So we're currently working in collaboration with Christos Prokakis from the UCL, to validate some of these mosaic structural variants. Finally, we have started like what I call a cancer pipeline. So it's basically merging many of the features of Sniffles 2 together. In this case, here's a tumor-normal comparison, and we can detect structural variants that are unique for the tumor when compared to normal, but not only for germline structural variants, but also for mosaic ones. So right now, we are this pipeline is kind of divided in two, either run for germline or for mosaic, but we're working on that you can just need to run it once, and you will get all the structural variants for germline and mosaic in a single file. So you don't need to compare like several files at the same time. So Sniffles 2 is good and everything, but there are some cases that were still missed. And this is basically on how how Sniffles 2 was developed, and it detects the structural variants based on the reads splits. So if you don't have a split, then you cannot detect a structural variant. And this is what happened here. One of the samples from Stanford. This is 108. So there was a terminal duplication that was not detected because the second breakpoint is not there. So Sniffles 2 cannot detect it. So for that, we have developed now a Spectra, which is a copy number variant caller that uses coverage in order to detect. And here, for this sample, we were able to easily detect this terminal deletion on chromosome 10. So we have applied this for the samples that I mentioned you before that were like Mendelian cases. For some terminal, we have here detect some terminal duplications in chromosome X. So it's based on coverage analysis. So we use MOSEF to get the coverage. And then we get like candidate selection of copy number variants, and then we remove some regions, like blacklisted regions, like Central American, Telemeric regions, or regions that are not present in the reference genome, for example, here using GRCH38. And then we merge contiguous candidates and do some filtering and statistics in order to detect the copy number variants, like here and here. So here, for example, here are two males that had a duplication, terminal duplication on chromosome X. We are using also SNV data, if available, to filter and for quality filtering, and we are testing loss of heterozygosity also. So in conclusions, with Sniffles and Spectra, we are covering the whole range of large structural variants. Both enable automatic comparison between samples, either tumor normal or population, and they are both available. I would like to thank all collaborators and people from my lab, and you for your attention. Thank you.